Doop, doop, doo. Good morning, everybody. I just got to pour myself a cup of coffee. Brain cells firing again. Doop, doo. I had a donut this morning. How cool is that? So I'm all good for sugar, at least. Caffeine, not so much. Boop-a-doo. All right, good morning to you too, Bianca. Let's see where we were at. Boop-a-doo. I can see what my schedule looks like as well. Much less interesting. Stir my coffee. Okay, whew. Ready to begin. Okay, so recap from, get this donut off my desk. Last class. Sorry, it's Friday and I haven't had any coffee. So essentially what we've covered so far is the kind of initiation process, right? And solution of some of the problems, right? So actually, why don't I ask you lot? What things have to happen and what problems does it cause in the initiation of replication, DNA replication? What happens first? This also gives me a chance to drink my coffee. What happens first in DNA replication? Yeah, think of this as like a mini study session. All right, yeah, it's either DNA gets, probably the better term, you're right, is it's twisted apart so that it forms single-stranded DNA, right? So you are right, and Stephanie too, right? So helicase can get in there. What else has to get in there to stop it from just twisting back again, right? Because remember, it's being untwisted against its will. So what's used to prevent it from just twisting back up again and forming double-stranded DNA again? There are other proteins required. They're actually on the picture in front of you, which is kind of funny. Helicase is one of the proteins that's required for expanding that bubble of single-stranded DNA. You're right, Isaiah. What are the proteins that stop it from collapsing back, right, before helicase and other things have started doing their job? It's on the image in front of you. Near to helicase, right? Look at what's bound to the single-stranded DNA. Single strand what? You're correct. Yeah, good stuff. Exactly. Single-strand binding proteins. Those physically prevent this DNA here from simply just collapsing back around the helicase and closing up. And they're constantly being pushed off by the DNA polymerase. Okay, so what's the role of helicase? What's its job? What does helicase do? You can speak as well. You don't have to type everything out. Might be quicker. Something to do with the helix, obviously. That's why it's called helicase. What job does helicase do once the initiator proteins have popped open the DNA? Single-stranded proteins hold it open. What does helicase do now to that small bubble, single-stranded DNA? Right, it expands it, keeps unwinding the DNA so that that bubble expands, and now we can fit all the other stuff in to that space. Right, so helicase is always, it's kind of like it's like the plow, right? Like I wouldn't say snow plow because many of you, oh, actually, some of you have seen snow now. Snow does happen occasionally in San Antonio, but we do not have snow plows. It's like a bow wave of a boat, just kind of pushing its way through, opening up the DNA. Now, what problem does that cause that needs fixing? It's all well and good, you're all correct, but what problem does that create? The helicase unwinding the DNA. Supercoiling, exactly. And what fixes supercoiling? DNA gyrase, topoisomerase, essentially the same thing. Exactly. And how? There you go. How does it fix the supercoiling? I think we've covered this, but I'm not 100% sure. I did check, but I didn't check well enough to know. How does DNA gyrase? Yeah, good stuff, Stephanie. Exactly. It essentially breaks the DNA and lets it kind of not unravel, but it's like I don't know if you ever tried reloading a weed whacker strimmer spool or winding up a hose or something like that, right? At some point, or even whenever I get my headphones out of my bag, you have to kind of like dangle it and let the cord spin a little bit so it's relieves that coiling. And that's essentially what DNA gyrase or topoisomerase does. Awesome, good stuff. So that should all be literally on the tip of your tongue. If you're still a little rusty, that's fine, but brush up on this stuff for the exam next week. Also, so I've talked about this already, I think, because we're at this tipping point, right, between the kind of more math heavy probability and statistics type stuff and the more process. Oh, that's a good question, Galen. Give me just a second. There will be a bunch of questions on like what order do these events occur in? What proteins do what things do these proteins require to do their job, and so on and so forth, right? So there's a lot of kind of fact-based stuff in a fairies. So how do they discover this? Well, that's a very good question. Probably the easiest answer, which is really how geneticists and molecular biologists discover most stuff, is they basically screw things up in terms of DNA and see what doesn't work anymore. Right. So if you mutagenize bacteria or yeast or stuff like that, which are the main kind of preliminary model organisms to this stuff, and one of those mutations happens to occur in helicase gene, then you may get slower DNA replication and growth, and that will start you on the path of, well, how does this work? This is a protein, we haven't seen it before. Get rid of it and DNA replication slows down, or rather, not get rid of it because then the bacterial cell will die, but reduce its function. Right? And then once you start with that, then you can do all kinds of things. You can see what proteins does this protein associate with, right? What other parts of this protein are required for this? You can do DNA binding assays, right? Where you basically just incubate the protein that you've purified, right? So you can work with it, and you look to see whether it sticks to DNA or not, right? And basically by doing so, it protects that DNA from being digested. Oh, X-ray crystallography is the answer to that question. So they will purify that protein. That's actually how the structure of DNA was discovered, right? It's actually pretty funky. Oops. Gak, get out of it. Sorry, I got all kinds of things blocking my DNA X-ray. Oh, I hate typing this out. Crystallography. Yeah, it's not quite typed well. Images. And there's Rosalind Franklin. So essentially, what you do is you make a crystal of this stuff, and then you fire X-rays through it, because X-rays have a very, 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 very short wavelength, which is how they pass through you. Essentially, they're able to, their wavelength is shorter than the distance between atoms. And so they can get bent as they pass through something. That's why you see bones, basically, when you use an X-ray. Now, I'm probably going to spell this wrong again. I'll show you, shoot, let's. Oh, that's for membrane proteins. Membrane proteins are a solid pain in the ass. I don't have any pictures of it. Maybe that's just because it was. There you go. This is a nice kind of image of the process. So you make a crystal purifier. That's why doing membrane bound proteins is really, really hard, because you have to crystallize them with membrane present as well. And then you shoot X-rays through it. Then you basically work out where the electrons are, because those are what help diffract the model, or diffract the X-rays. Then you get an atomic model based on that, which is essentially done by very, 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 very clever people, because, I mean, this is whoosh, straight over my head. But really the cool thing about all of this, and this is why I'm kind of chunning on about it a bit, is that these are all essentially, scientific knowledge is not a linear process, right? A lot of people think that you discover this, then this, then this, and then this, and then you get to your destination, whatever that might be, the fact that you're talking about. But really, scientific knowledge is more like a pyramid, like the, shoot, I've forgotten the name of those pyramids in Mexico, some outside Mexico City. It's a word that I can't pronounce very easily, tear something or other. Anyway, it's more like a pyramid, right? All of these different bits of information are kind of the foundation, essentially, right? And they're built upon by other people who take that knowledge, like the mutation of a helicase, and then they apply that to their particular, speciality, right? X-ray crystallography, for example, right? And then that's then used by, molecular geneticists who look at this atomic model and go, ah, that looks like an active site. Wonder what happens if I mutate this particular amino acid, change it. Does that active site still work, right? And then that kind of, it all builds up together until at the very top, you get what I'm teaching you, essentially. Or actually what I'm teaching you is kind of like, I don't know, 2 or 3 layers or levels below the top, right? The top is where people stand around going, wow, wonder where this is going, right? What you're learning in college, at least at this level, is stuff that has been figured out, and has been figured out for varying amounts of time. Teotihuacan, that's it. I've been there. It's a really cool place. It takes a lot of effort to get up one of them. Thank you, Melody. It's a very, very neat place. But anyway, so, this is kind of known stuff, right? It's been worked out pretty thoroughly already. As you go higher up in your degree, or even a postgraduate degree, then you start getting to the top of that pyramid until you're the one that's actually adding, bricks to, or stones or whatever, to push the analogy further, to raise that pyramid even more, right? So at some point, if you continue on into postgraduate stuff and you're the actual person making these discoveries, you're the one that knows the most about this particular thing in the world, typically. And there is stuff that is not known, right? So quite often people ask you questions, I'm like, I don't know. And I don't know whether it's because I'm ignorant, right? I don't know it, or if it is because it's not known, right? In which case no one knows it, right? So it's sometimes hard to give a good answer because of that. Well, yeah, you know, research and teaching are, you know, they're either side, two sides of a scale, right? Some people have a more emphasis on one, more emphasis on the other, but they do interact. I've learned an enormous amount of stuff from teaching. And that stuff that I've learned has actually informed my research as well. So it goes both ways. Anyway, that was a little bit of a tangent, but thank you, Caitlin. That's a good question. So yeah, there's an awful lot of different avenues for investigation to discover this thing, these kinds of things. And actually, like I guess, most recent kind of amazing discovery in molecular biology, which is CRISPR-Cas9, which we'll talk about a little bit later on at the end of the course. That was discovered by looking at the natural immune defenses of bacteria, right? It wasn't something that scientists went, I'm going to go look for an awesome transgenic method. No, it's just people who are working on, shoot, I can even tell you what bacteria, Streptomyces, I think. No, that's a yeast. Yeah, I don't know. Anyway, they were just interested in it as a biological mechanism. And it's basically turned into this, I don't know, almost Pandora's box where we can now technically genetically modify humans, which is a little scary. Anyway, all right, fine. Before I get distracted further, which is quite possible. Next problem. And there you go, next problem. I don't know what's going on with my trackpad. Anyway, so here's another bit of revision. Don't you just love me today? All this revision. Hopefully I balance it, right? You know, not so interesting stuff, interesting stuff. So what do you need? What does DNA polymerase need in order to add a nucleotide to the daughter strand? Right, talked about this a bunch. Oh, Stephanie, shush, just for a little bit. Or actually, let's expand upon that. What does RNA provide that DNA polymerase needs? Where does a new nucleotide get added, whether it's RNA or DNA? And I'm looking for a number. I'm looking for a couple of letters. So, first word, 2 syllables, 1 number. Hey, charades in genetics. Did that actually help, Daniel, or are you just thinking about that anyway? Yeah, 3 prime. 3 prime what? Awesome. Well, you know, whatever it takes, right? Looking for 1 word, 2 letters. Or 1 word, 3 syllables. 3 prime what? Yeah, OH. That was the 2 letters one. Or hydroxyl group. Ah, shit, that was 2 words. I'm not very good at playing charades. Yeah, so you need a 3 prime OH, right? 3 prime means that OH is attached to what in the existing nucleotide? What does the number refer to? Begins with a C. Oh, damn it. It's actually kind of telling the answer. Yeah. Thank you, Armando. You saved me from even worse attempts at kind of goofing around. Right, 3' carbon, right? Because that 3' carbon has a hydroxyl group attached to it. And that is where a new nucleotide will be added. So where's our 3' carbon? Or 3' OH, either one. Actually, 3' OH is probably better. Where's our free hydroxyl group in that image? Bit of a trick question. It is at the end of this strand, unless it's circular, right? But it's not here. Yeah, there isn't one, right? There isn't one because none has been added yet. And DNA polymerase cannot take a nucleotide, a DNA nucleotide, and just plonk it down here opposite its complementary base. So now we get back to what Stephanie wrote is you need some RNA, right? And this is called a primer. So RNA poly or polymerase, oops, quite happily adds RNA nucleotides to a complementary DNA strand. Basically that equals a DNA RNA hybrid. Because that's what RNA polymerase does when it transcribes DNA and makes mRNA, right? It doesn't have any issue doing that. And so what we need now is what's called a primer. You need an RNA primer. And this is pretty short. I don't know, probably 15 to 20 base pairs or nucleotides long. But what it does, and if you kind of just hold up your hand, you know, and kind of block out the, what color that is, pinkish bit, I guess, which is the DNA. At the end of that RNA fragment, you will have a 3' OH. Oops. Oh, I don't know where I was typing. Primer provides the 3' OH, PH, OH. DNA pol requires. So as soon as that OH is there, you're off at the races. DNA polymerase comes in, starts synthesizing quite happily, right? Now, another question, get you think about things. Per replication bubble, let's say we just have unidirectional replication, right? Just to keep things simple. How many primers, RNA primers, per replication bubble, or even per replication fork, actually, would you expect for the leading strand? How many do you think you'll need? What is the characteristic of the leading strand? Yeah, just one, right? Because it's synthesized continuously. So you prime it once, off it goes. How many do you think will be needed for the lagging strand? Just one or lots? Yeah, you're going to need one for every single fragment. Every single fragment, right? And so that is why the lagging strand is the lagging strand. Because it has so many RNA primers in it that need to be removed. Synthesis, or the replication, occurs at the same rate as the leading strand. Tidying up all that mess that's created takes a lot longer. So that's just kind of quite nicely shown here also as well in the original picture here as well, right? You have multiple primers created for the lagging strand. You can't even see the one for the leading strand because it's like way over there. Again, just because it's important, you only add a new nucleotide to the 3' end using that OH. Now, this is there's a little bit here that's worth kind of pointing out. One is that this hydrogen bonding and the size is what ensures a reasonable degree of replication fidelity, right, or accuracy. It's part of, but not the whole part of why we don't get a huge amount of mutations every single time we copy our DNA, which is thousands of times over your lifespan from being a zygote to being an old man or a woman. Second thing as well is that this bond formation, this phosphodiester bond, is catalyzed by the energy in this phosphate-phosphate bond. So incoming nucleotides have 3 phosphates, very much like ATP, right? They are very, very similar molecules, not the same, but similar. And just as with ATP, the energy there is between the phosphate-phosphate groups. Different bonds between the two, but it's really all about the energy. This is important later on when we tidy up all of this mess. Right, so as part of that bond formation, you release this diphosphate group called a pyrophosphate. Prokaryotes are relatively straightforward. They don't have many DNA polymerases. They only have 5. And they're called 1 through 5 simply in the order in which they were discovered. Right, there's a lot of weird stuff like that in biology. It's just like, oh, we found something cool. We're going to call it something number 1. And then you realize it actually acts later than something else, but it's already been called something number 1. So that's just what you stick with. So pol 3 is the one that does the bulk of the DNA extension. This is going to be a little bit different in eukaryotes. Eukaryotes do it a little bit, yeah, funkily, right, compared with prokaryotes. There's another DNA pol 1, which we'll get to in a second, and the rest are involved in DNA repair, which we'll also cover maybe before the end of class today, if I avoid getting too distracted by other stuff. So basically, this lagging strand has a lot of work that needs doing to it. It essentially exists as a series of small fragments. So if it was left as that, you'd have 2 major issues. One, you'd have RNA in your genome, and you don't want that, right? Because if that happens to be in a coding sequence, well, you know, RNA polymerase is going to come along and go, don't like the look of that, and you're not going to get that gene transcribed. Right? The second major problem, and this is in many ways an even bigger one than the RNA being there, is that the phosphate sugar backbone is broken. It is not continuous. It's kind of like I always talk about railways for some reason, don't know why. It's kind of like the analogy of laying track and you don't bother joining the rails together. You just kind of like plonk them down and go, that looks good. Obviously, that's not going to work very well, right? So before DNA replication can be considered complete, the RNA primers have to be removed. The bit of space, right, that doing so creates, right, because essentially you've moved part of the daughter strand, that needs to be filled in. That's what DNA pol 1 does. And then last job, you have to reseal the backbone, right? You have to create a phosphodiester bond between the end of one fragment and the beginning of the next one. I always refer to this as clearing up the mess, right? Because all of these Okazaki fragments are basically a solution to the anti-parallel DNA problem. Now we have to fix the problem that creates, essentially. And so DNA pol 1 is it's a pretty cool enzyme. It has two domains, like parts of the protein, right, which do very different things. One is a RNase, and so an ASE is an enzyme essentially, and RNase is an enzyme that destroys RNA, right? And it recognizes there you go. A DNA RNA hybrids and choose up. Okay. Don't know what's going on with my type. Choose up you? No. Choose up the RNA. Right, so that's one thing that it does. Second is DNA polymerase domain that basically continues synthesis of the fragment. So as the RNA nucleotides are being kind of broken up, like hydrolysis of those phosphodiester bonds that hold the RNA in place, as it's doing that, kind of like in front of it, behind it, it's putting in the correct DNA nucleotides to fill in the gap that removing the RNA creates. So by the time DNA pol 1 is done, essentially you have every base on the template strand has a complementary base in the daughter strand. DNA synthesis is not finished yet because we still have this break in the phosphate sugar backbone. Also kind of cool as well is DNA polymerase 1 sucks at long distance work. It is really, really optimized for short, small bits of DNA. That's why DNA pol 3 is the main workhorse that does the long distance stuff. And DNA pol 1 kind of does the kind of those short, dirty jobs. It's like the difference between Gordon and Thomas the Tank Engine and one of those little yard shunters, I guess. My kids were nuts, particularly my eldest was nuts about Thomas the Tank Engine. I can actually hear the theme song going through my head right now, which is a little disturbing. Haven't heard that for a long time. At least it's not Peppa Pig. That one annoyed the crap out of me. So anyway, now we need to fix that nick, right? We need to create that phosphate that phosphodiester bond between the end of one fragment and the beginning of the next fragment. So what we need now is another enzyme, and that's DNA ligase. And ligate basically means to stitch, if I can spell it right, stitch together. Like ligature is a fancy word for a piece of thread that you use to sew up a wound. What else? Ligand? There's lots of lig words, right? But anyway, all bases from that, stitching stuff or sticking stuff together. But it also requires the energy in ATP. And those two together creates the phosphodiester bond between the two fragments. And the main reason for needing that ATP is because essentially the energy in those phosphate bonds aren't available anymore, right, in the original nucleotide. It just doesn't work the same way. And so you need to bring in energy to carry out that chemical reaction, and that energy is in the form of ATP. So if you're ever to do one of these, right, in a lab, you would get your ligase, right, little tube, and you'd also have to get some ATP, right, which you can also get in a little tube, and add that. Otherwise, the whole thing will not work. All right, anybody got questions so far? There will be questions on this order as well. Nope. Well, if you do, just shout them out. And then it ends. And usually it ends when you have two replication bubbles kind of merge, right? So as these expand and DNA is made, eventually, if you're bacterium, those two replication forks, or even one, will come round and it will hit, you know, the other side of another one or the same one. If it's a eukaryote, those replication forks move outwards from the origin of replication until they hit other replication forks moving towards them from, again, other replication origins. Sometimes though you do get actual termination proteins. That's kind of fairly rare though. Now, DNA replication can introduce errors, right? And this is why many of the proteins involved in cell cycle checkpoints, which we learned about a little while ago, are involved in either detecting errors in DNA or problems with DNA, or involved in fixing them. And that's the DNA repair side of things is actually the main role of the BRCA1 and 2 proteins, which when they're absent or non-functioning can increase the possibility of breast cancer, right? So essentially what happens is occasionally, you know, just by error, I'm not entirely sure, just stochastically, I guess, the wrong base makes its way into the DNA polymerase and somehow sticks, right? Shouldn't do, but it does. And so this is called a mismatch, right? There should not be a guanine base paired with an adenine, right? It should be a thymine instead. But DNA polymerase can't see what should be there, right? It's not like it's reading the DNA. It's just bases are coming in, and if they match, then a phosphodiester bond is formed. So this phosphide ester bond gets formed, and then the DNA polymerase basically goes, ooh, shucks. Think of it like some corn farmer in Oklahoma. That ain't no good, or whatever Oklahoman farmers say. I don't know. And it stops, and it essentially backs up a little bit. And what it does is it cuts that phosphodiester bond itself, right? So it has what's called exonuclease activity. And exo means from the outside, which is the opposite of endo, which is from the inside. So there are endonucleases as well. And so it's able to cut off the offending nucleotide, and I don't actually know how it senses it. I would imagine it does so based on size or shape of DNA, right? Because these two, the distance between these two strands is not going to be correct. Either it'll be a little bit too small, right? If you have two pyrimidines stuck together, or in the case of this case, this is two purines, right? So there's now 4 rings between these two bases. So it's going to push the DNA out a bit. And my guess, I don't know for sure, but my guess is that the DNA polymerase senses that, and that's what makes it stop and then cut off that offending base. Essentially, then goes, whew, fix that problem, and it starts again, right? And it just kind of continues onwards. Now, the issue is, once another base has been added, right? Let's say we had another guanine, which is the correct match for cytosine. It can no longer fix the problem because its exonuclease activity can only act on the last nucleotide, the end, essentially, of the DNA strand. So if a mismatch occurs and it's not fixed, then essentially that change could possibly end up being incorporated, which would not be good. We don't want mutations, right? Mutations, if they have any effect at all, which they don't always do, but generally if they do have an effect, are bad, right? Even though, you know, we are the result of essentially millions of mutations and we end up the way we are as a result, that's a tiny, tiny, tiny fraction of the mutations that have been experienced by our evolutionary ancestors, right? Most of them either did nothing or resulted in bad things happening for that individual. So typically, I think the error rate for DNA polymerases without exonuclease activity, and you can get them, like for some reason they're really cheap actually to buy them for molecular biology, is about 1 in 1,000, give or take, roughly speaking. And so this proofreading mechanism where the DNA pol goes back and fixes its own mistake, that increases that to about 1 in 10,000, about 10 to the 5 accuracy. Where are we at? Come on. Yeah, about 10 to the 5. So that'd be basically 1 error in 10,000 bases on average, right? But that still kind of sucks, really, right? If you consider a bacterium has, I don't know, genome of in the millions of base pairs, you're looking at hundreds of mutations per, or tens to hundreds of mutations per round of division. Bacteria replicate really quickly. If that was in a multicellular organism, you know, our genome is 6 billion nucleotides. You would have thousands upon thousands. I think I worked out like hundreds of thousands of mutations every time your cells divide. And that would be pretty much catastrophic, right? You'd end up with the cells at the end having a completely different genome to the ones at the beginning. And so there's extra stuff that goes on. And essentially what happens here is, say that error did not get caught by DNA polymerase. Now we need to catch that error really quickly, right? So DNA polymerase has shunted off into the distance, doing its job, oblivious to the problem that it's caused. We have to detect and fix that error before essentially the DNA is made to look, the new freshly made DNA ends up looking like the old template DNA, right? And essentially, and this is a cool thing which we'll talk about a bunch when we talk about epigenetics. DNA is methylated on C's or A's. And that basically means some bases have methyl groups on them. And that changes what they look like, right? It's actually a big part of how transcription is controlled and also how cancers can progress as well, based on differential methylation. But the point in this case is that methylation doesn't happen for a short time after DNA replication proceeds. So for a gap, there's like a window of opportunity here. You can tell which is the old and which is the new, because the old is methylated, the new is not. So if you find an error, a mismatch, now you can go, well, this strand is good because it has methyl groups. This strand, this one's bad, right? Because there's a difference and this is the new group. So the difference must be in the newly synthesized DNA, right? That little window only stays open for a short time, right? Because the methylation occurs, after that replication fork has gone off into the distance. And so essentially that mismatch is recognized again by shape, right? You'll literally get a little bubble in DNA where these two bases will not stick together properly. And so that's recognized by, in bacteria, a bunch of proteins called MUT or mute proteins. And what they do is instead of just cutting out the one offending nucleotide, they destroy a whole patch of the daughter strand. And so what they do is they'll scan along from that mismatch until they find the next methylation point, right, in the template strand. And then they'll just destroy all of that part of the daughter strand. So essentially creating single-stranded DNA again. Then once that occurs, now DNA polymerase, one of those DNA repair polymerases, comes in and redoes the job carefully, one would hope. And the same deal again, DNA ligase will seal the backbone, and then after a period of time, DNA methylases will come along and fix that. So essentially, DNA replication really doesn't kind of finish until all of this has occurred. Right? Actually, I got my numbers a little bit wrong. The inherent accuracy of DNA polymerase is about 10 to the 5, 10 to the 4 to 10 to the 5. The proofreading improves that by 2 orders of magnitude. And then the mismatch repair improves it further. So essentially, overall, the error rate is about 1 in every billion to 1 in every 10 billion nucleotides. Yeah, I mean, that's natural selection and evolution for you, Caitlin. Because if you think about it, there's a very strong selective advantage to not having your DNA mutate in ways that don't benefit you, right? And so there's all of these proteins that do these jobs exist partly through accident, but also because those accidental changes, whether they're gene duplications or mutations or whatever, actually improve the fitness of that organism. So that's kind of what I mean in a sense, like what we observe now is the end point of a very, very long process. And basically what you need to do in terms of evolution is stop, turn around and look backwards, right? And our last common ancestor with bacteria would have been, oh, when would that? When did eukaryotes? I think that's probably about 2 billion years ago. And they think, not very sure, right, because it's hard to tell, but they think life emerged on Earth between 3.5 and 3.8 billion years ago, which isn't that much younger than the Earth itself, which is around 4.5 billion years old. So that's just a very, 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 very long time, and millions upon millions of generations where essentially all this stuff gets refined until we get to the point where we go, this is what we look like. So that's kind of essentially how, I mean, it's just really, really cool. It's, yeah, biology is neat. I like it. That's an interesting question, Isaiah. So, I mean, probably to some extent, it depends on what methods are used to change that DNA. I imagine that there will be signatures that result from doing so. Even CRISPR-Cas doesn't necessarily leave a signature, but it occurs in a particular way that allows you to tell if that mutation was introduced synthetically, essentially. But yeah. Oh, and we're kind of off the reservation already. But if you haven't already, you should watch a film called Gattaca. I should really make this some kind of homework. And that's basically a super cool, kind of old. It's got shoot, I can never remember his name. And the actress out of Kill Bill, can't remember her name either. I'm getting old. It's kind of depressing. Anyway, about the essentially the societal consequences of genetic modification. Uma Thurman, there you go, Amanda. Thank you for that. I should remember that stuff, but you know, I guess my brain's full of other stuff. Things just fall out. Okay, cool. Anyway, I've gone over time. That's it from me today. I hope you have a great weekend. On Monday, we will just summarize this stuff and then we'll get onto homologous recombination, which is a pretty wild kind of process. All right. See you all soon. Have a good weekend too.